Okay, so this week is week 13, Circulation and Diving Physiology, chapters 25 and 26 from the textbook. Before we start on the material, I wanted to say something about the Top Hat grading. Hopefully you all have seen my email about the challenges that we've had with Top Hat. Basically, what happened was that before the semester started, I tested the system, and when there was a page, as I've been making for each week, with multiple questions embedded, it was possible for me to get the output of each, that is a report for each page, with every question itemized. So for every question, I got a score for correctness and for participation. After the semester started, very early in the semester, Top Hat updated their software and removed that functionality. So now with the page, the only report that I can get is a composite score of all the questions and discussions in terms of correctness and participation, which is not useful because I hadn't planned on using all of those points. So as I mentioned in the email, basically the Top Hat grading has not been working, so that's why you haven't seen a grade for your Top Hat scores. But what I can tell you is that if you are in general using the Top Hat system, submitting your questions, and basically doing the work that I've asked you to do each week during the lecture, you will get a good score on your Top Hat. So you can expect that in your final grade. I'm trying to work with Top Hat to get them to extract the scores for me the way I want. Basically, there's a way to do it, but it's a very laborious. I do not have time to do it, and so I've asked them to do it for me. But whether or not they actually provide that service, I do not know. I apologize. They should apologize. You could write them angry messages if you want, telling them that you don't like the fact that they changed their software during the semester, which I have also told them. But it is what it is, and I want you to not worry. You will get a score in Top Hat that's essentially in the A range if you have been doing all the work judiciously, in the B range if you've done some of the work but not all of it. That is what I can tell you about that at the moment. Okay, so let's look at the week 13 material. So for starters, we're going to talk about circulatory systems. And almost all animals have circulatory systems, but there's some that don't. Sponges don't have circulatory systems. Cnidarians, like corals and anemones, they do not have circulatory systems. And flatworms, the platyhelminths, don't have circulatory systems. In the case of sponges, their body sizes can be very large. And they actually do circulate water through their bodies. There's a cool video on Top Hat for you to watch of a sponge where glowing dye called fluorescein is put on the outside of the sponge, and you can see the sponge takes this dye inside of its body and out the big hole on the top, which is called the osculum. And so sponges do this because they filter the water and they catch the food that's in the water, and they eat that. It's not really a circulatory system in that fluids are not passing within the tissues of their body because they don't have tissues. But the fluids are definitely passing past their cells, especially these choanocytes, where the cilia are beating to propel fluids through. Cnidarians do take water inside of their gastrovascular cavity. And cnidarians can use this to swell or shrink. You may have especially noticed this in sea anemones, and of course, the mouth is also the anus in a cnidarian, and they only have two tissue layers, endoderm and ectoderm. They don't have any mesoderm. And so they use muscles to basically open or close the mouth and take water in or expel water. And then flatworms, well, they're flat. And they basically can take advantage of diffusion distances that are very small between the external and the internal environment. But for all the other animals, they do have some form of circulatory system. And we can think of the circulatory system basically as two parts, the pumps and the pipes. So yes, this is just like a plumbing problem. The pumps are the main propulsive organ, which is in general the heart that forces blood throughout the body, but we'll see that there are some other pumps as well, especially involved in returning blood to the heart. And so that's the main propulsive organism. And this is where we get the cardio in cardiovascular. It's from the cardiac or the cardio pump. And then there's the pipes. So the pipes are made up of an arterial system, capillaries, and a venous system. So the arterial system, those are the pipes that take blood from the heart and distribute them to the tissues. And the arterial system is high pressure. So, so it can pressurize the blood to force it through very small spaces, which can be the capillaries. The capillaries are really only present in organisms that have closed circulatory systems, as we'll talk about. And these are very small blood vessels where materials are transferred between the blood and the tissue. And then finally, the venous system. The venous system is basically a blood storage reservoir. It's very expandable. It has very low pressure, and this is where blood gets returned to the heart under low pressure so that it can be pumped out again. So the pump is the cardio, the pipes are the peripheral circulation, and this is where the vascular part of cardiovascular comes in. Okay, so open and closed circulatory systems. In an open circulatory system, there's some sort of pump, like the heart, and then there's some degree of arteries. It can be very small or relatively large, but at some point, the artery is just open. And blood, or actually hemolymph, in the case of an open circulatory system, we don't call it blood when it's an open circulatory system, we call it hemolymph. Hemolymph flows out into a space called the hemocoel, which is basically just space or sinuses around tissues. And there, the hemolymph makes contact with the tissues. And eventually, it flows into either more or less well-developed venous system that returns the hemolymph to the heart so it can be pumped around again. So this is an open circulatory system. In a closed circulatory system, in which we call the circulatory fluid blood, there's no escape. The blood does not escape from the circulatory system, except as we'll see in the case of injury or specialized kinds of capillaries. But basically, the heart pumps out into an arterial system, and then there's a series of branches into smaller vessels called arterioles, the smallest vessels called capillaries, larger vessels called venules, and then into the largest veins and get returned back to the heart. And so it's at these capillaries where the contact with the tissues is made. Okay, so the cardiac pump, or the heart, has a rhythm of contraction and relaxation. So of course the heart contracts when blood is pumped out, and it relaxes when it fills up again, so it can replenish the blood and pump it out again. That period of pumping or contraction is called systole, and the relaxation or filling is called diastole. There's a top hat video of a human heart beating. It's pretty interesting to watch, and you can see what it looks like when the muscles are contracting and relaxing in systole and diastole. The control of this rhythm of pumping is by special cells called pacemakers. So pacemakers are cells that have a rhythmic depolarization, repolarization cycle, and that natural cycling in the pacemaker cells is what sets the rhythm of the contraction of the heart muscle. And the pacemakers differ across different animal groups. So in vertebrate animals, the pacemaker is myogenic. So that means that the contraction of the heart initiates in specialized non-contractile cardiac muscle cells. So they're actually muscle cells that don't contract, but send electrical impulses across the heart to make the heart muscles contract. And so we'll talk about that a little more detail. Invertebrates have pacemakers that can be either neurogenic, that is, they're actually neurons, or myogenic. So the neurogenic pacemakers, for example, are nerves, specialized nerve cells that innervate skeletal muscle cells, or they have the neuromuscular junction with skeletal muscle cells, and the heart is made up of skeletal muscle cells in that case. So we're going to look at the circulatory systems of different animal groups. Of course, in annelids, there are three main classes: the polychaetes, the oligochaetes, and the leeches, the Hirudinea, and they have different types of circulatory systems. In the polychaetes and oligochaetes, they have open circulatory system, and move. The polychaetes have an open circulatory system, and so they have a heart, and they have artery systems, then they have sinuses or spaces around their bodies. Oligochaetes, like the earthworm, actually has a closed circulatory system, and they have a number of hearts up in the anterior part of their body, and then they have a sort of paired arterial system that runs down the length of their body. And every segment of the body of an annelid has a connecting vessel, basically going from the one side of the body to the other. So the open circulatory system in polychaetes, a closed circulatory system in oligochaetes, and here their hearts do some of the pumping of fluid, but contraction of the muscles of their body also are important for this purpose. In arthropods, there's a lot of variation. So all arthropods have one or more hearts and some blood vessels or hemolymph carrying vessels, and all of them have open circulatory systems. But in crustaceans, the circulatory systems become more complex, especially in large crustaceans. So for example, here's a branchiopod crustacean, a fairy shrimp, and the fairy shrimp has basically one long tube that is its circulatory system, and there's contractions happening along every length of this tube with openings in the tube called ostia. And so as the tube contracts, basically serving as the heart, fluids are forced out of the ostia, and when the tube relaxes, fluids flow in, or hemolymph flows into the ostia, each of the ostia. In the decapod crustacean, like the crayfish or lobster or crab, there is one big heart, which is kind of under the carapace here, and the heart has several ostia in it. And as the heart contracts, hemolymph is forced out through a fairly well-developed arterial system that directs the hemolymph to various parts of the body. And actually, the vasculature of the crustaceans, the larger crustaceans, can be very complex, and these arterial systems basically continue into what looks very much like a capillary bed, but then they have an open venous system, and the hemolymph basically returns to the cardiac chamber and flows into these ostia. So here's another picture of a crayfish or a lobster, showing you all of the various arteries. So there's the heart is here. There's a very large dorsal abdominal artery. There's a very large sternal artery that goes down to the sternal part of the animal. There is this ophthalmic artery that carries blood to the brain and to the eyes. Hemolymph to the brain and to the eyes, etc. So there's quite a lot of vasculature here, and actually it can continue out into the legs and the claws. So if we look at the circulatory plan down here in the lower left of the larger decapod crustacean, we see that the heart here pumps hemolymph out into the arteries and eventually into the systemic tissues, where sort of in this region here, that's the open part of the circulatory system. And then there are venous-like systems that collect the hemolymph and pass it over the gills before there are basically veins. So they're called branchiopericardial veins, or gill near the heart veins that carry the hemolymph from the gills back into the heart. And so basically when the hemolymph is around the muscle tissue or some of the other tissues, it's not inside a vessel, but it starts becoming inside of a vessel very close to the entry point into the gills. The crustaceans have a neurogenic heart. So this is another picture of the heart right here, showing the major arteries coming out of the heart. And here are the ostia, or each of the ostia, that are the openings where hemolymph flows into the heart. And the heart is sitting under sort of a muscular membrane here that has various attachment points to different parts of the skeleton of the animal. And so as the heart contracts, of course, these ostia are closed, and when the heart is relaxed, the ostia are open, and hemolymph flows in through the ostia. Here's another picture of the heart. So this big yellowish part here is the heart. And here are all of the pacemaker neurons that are on top of this heart. And there's 9 different neurons. There's 4 over here that are small cells, neurons 6, 7, 8, and 9. And the small cells initiate the contraction and, or initiate the depolarization, I should say. And then that depolarization spreads from the small cells into these larger cells over here. And the larger cells are the ones that are actually the motor neurons. And so you can see, like, here's the motor neuron synapse points. And so they connect across the heart muscle, and they depolarize rhythmically so that all of the heart muscle contracts all at once. Because the key here is that the whole thing has to contract all at once, and the whole thing has to relax all at once. So these neurons are intimately connected to the heart, like they're part of the heart. They're sitting on top of the heart muscle, and it's these small cells that initiate the rhythmic contraction and relaxation cycles of the larger motor neurons. Insects also have hearts and an open circulatory system, but their circulatory system is not really supporting respiratory gas exchange. That happens through the tracheal system, as we talked about last time, gas-filled spaces inside the insect. Instead, the hearts of insects, which include contractile bodies within each segment, as well as in the base of the wings, are forcing hemolymph through the insect's body, probably for the purposes of like moving nutrients and taking waste products away, but not for the purposes of gas exchange. Mollusks have at least one and sometimes more than one heart, and they have blood vessels as well. Most mollusks have open circulatory systems, but cephalopod mollusks are special, and they have closed circulatory systems. So here's a gastropod snail right over here, and they have several. Okay, so the gastropods have a series of hearts that are involved in pumping blood. So right here is the large ventricle. So here's the gastropod. Here's its shell would be on the outside. Here's its foot. Here's its head. And the red colors are showing its vascular system. This is a land snail, so it has a lung and also has some gills. Basically, the circulatory plan for a marine snail is that there's more vasculature in the gills. And of course, for land snails that have a gas-filled space or a lung, there's vasculature around the outside, but not through the center. So the heart is the main propulsive muscle of the heart is the ventricle. And the ventricle is pumping blood out, or hemolymph out and into several places. One place is it's pumping it out into this expandable atrial organ here, so it's sort of like a secondary heart. And that helps to pump the blood up into the head space and through the foot, and it's also pumping the blood ventrally as well as dorsally. The blood returns to the heart basically by going through a series of tissues and digestive organs and respiratory structures into an accessory heart, which is here, this A sub U, and that is a contractile vessel that that basically expands and then contracts to fill up the ventricle. This little thing over here that is supposed to be an animation, but the animation doesn't work in Top Hat. In a bivalve mollusk, like a clam or an oyster, basically have a more open system. We have the heart is here, the main propulsive organ, and then we have arterial systems that are sending blood over to the mantle or into the foot tissue. So this is the foot right there of the clam. Here's the gill tissue here. And so we can see that the gills are just like the crustaceans, where there's an infrabrachial sinus that's here, and then a brachial vessel that brings the hemolymph back through the gills before it gets returned to the heart. Cephalopods have the most complex circulatory system of any mollusk, squid, and octopus. They have a closed circulatory system, and they actually have 3 hearts. They have a systemic heart and 2 branchial hearts. There's a cool Top Hat video of this also, where they put an endoscope inside the squid, and you can see the various hearts beating. The systemic heart is what's pumping fluid through the tissues, and then the deoxygenated blood goes into the branchial hearts, which are basically forcing the blood through the gills and then back to the systemic heart. So the gills are very large structures in cephalopods. They have very high metabolic rates, of course, and so they have to have a lot of gas exchange. And because the structures are so large, additional pressure is needed to force fluids through them. And so hence that's the purpose of these branchial hearts, is to force the fluids through the gills. Okay, here's another look at the same thing. Again, the systemic heart, the systemic tissues, the branchial hearts are here, and then the gills. Totally closed circulatory system. The systemic heart is here in the middle, and the branchial hearts are basically situated right under each of the sets of gills. The hearts, of course, need to have some degree of oxygen in which to function, and they are basically being perfused by venous blood. And so they actually are operating under relatively low oxygen compared to the systemic heart. And so these two types of heart muscle may have very different types of metabolic poise. Invertebrate chordates, for example, urochordates, the tunicates, and the cephalochordates. The lancelets are invertebrate chordates. Of course, vertebrates are also chordates. Tunicates have an open circulatory system with a tubular heart at the base of the digestive tract. Most tunicates are relatively small. Some of the big ones, Siona, might be, like 6 inches long. The cephalochordates, or the lancelets, which are all very small, have a closed circulatory system with a few open sinuses, and also a tubular heart at the base of the digestive tract, and blood vessels that are also able to contract and force blood through the body. Fish have hearts with one main propulsive organ, the ventricle, which is here, highly muscularized, and then they have some non-contractile chambers to the heart. So a fish heart looks like this, and there's also a top hat video of a fish heart that you can see. So the ventricle, this is the main propulsive organ. It's very dark red in color. It's the tissue is full of both myoglobin in the heart muscle as well as hemoglobin in the blood that is moving through it. So blood, there's a closed circulatory system. Blood returns to the heart through this main venous chamber, the sinus venosus, and fills up the atrium. The atrial chamber is non-contractile, and it's just sort of like an elastic tissue where when the force of blood fills it, it expands a little bit, and the pressure of that expansion forces the blood into the ventricle when it relaxes. And then when the ventricle contracts in systole, it forces the blood out into something called the bulbus arteriosus. The bulbus arteriosus is also non-contractile, but has huge elastic properties and is what sets the blood pressure of the fish. So this is kind of a main pressure reservoir. The fish heart is made up of something called spongy myocardium, which basically means that the muscle cells have lots of holes that run through them, kind of like a dish sponge. And the blood that's flowing through the heart flows through those spongy holes as well, and provides the muscle tissue with the oxygen and whatever sugars or amino acids that the heart muscles are using for metabolism. So different types of fish can modify their circulatory systems depending on the extent to which they use their gills or other types of gas exchange structures. So here's a typical water-breathing fish. Here's the heart down here, as we just talked about. And the heart pumps blood that is largely deoxygenated past the gills. So the first thing that happens is that the blood goes to the gills and becomes oxygenated at the gills through this afferent branchial artery. And then there's the efferent branchial artery that takes the blood from the gills. So this is, of course, a capillary bed here at the gills with very fine blood vessels. So there's a lot of pressure, then there's low pressure here, and then higher pressure leaving the gills through the dorsal aorta, the main arterial system, that then takes the blood into the bodies of the fish. There can be circulation that goes through the tissues. That's called primary circulation. And then there can be a secondary circulation that skips the tissues, and it has something called an arterial anastomosis. This is basically like where there's a short circuit. So some of the blood doesn't go to the tissues, some of the blood just kind of short circuits. And this is important in fish because if all of the blood went to the tissues, like the muscles and the liver and wherever else the blood is going, all the oxygen would get used up, and there would be no oxygenated blood that was getting back to the heart. But because of this arterial anastomosis, some of the blood that's oxygenated at the gills skips the rest of the body and basically goes right back to the heart. And in fact, let me see if I can find my colors. Nope. Colors. Oh yeah, there we go. So some of this blood is actually oxygenated because it skipped the tissues. And so the fish gets some oxygenated blood back to the heart, and that's important because the heart then has oxygen, which the muscle, of course, needs to beat. And so that's the whole purpose of this arterial anastomosis, is to be sure that there's oxygenated blood that makes it back into the heart so that it can go into the spongy myocardium and the heart can continue to beat. Okay, so so here's what we just talked about with an ordinary fish, with actually this anastomosis right here, and an electric eel has the same kind of thing, except an electric eel is an air-breathing fish. I don't know if you remember that electric eel video that we watched before. And it has a slightly different circulatory plan. Well, actually, it's the same circulatory plan, except here the gills are not functioning. So that the electric eel's vasculature past the gills aren't part of the respiratory exchange. Instead, the mouth is an air-breathing organ. So the electric eel basically gulps air into its mouth and uses that for breathing. And so a top hat question for you to address is, what are the negative consequences of the air-breathing organ, or the mouth in this case? And here's a picture of an electric eel with its eyes are there, and here's its mouth. Its mouth is closed, and when you open the mouth, which is this picture on the right, you basically see the surface of the mouth is very red, and that's because there's a lot of vasculature there. There's a lot of pipes for blood there. So what are the negative consequences of the air-breathing organ? So look over at these pictures right here and see what you think of as a negative consequence. And answer that question on Top Hat. So hopefully you answered this question, and what you saw was that the systemic tissues of the electric eel don't get as much oxygen as the systemic tissues of a branchially breathing fish, because the blood can never become fully oxygenated because of the fact that the air-breathing organ is happening before the blood gets back to the heart, and so the heart is using up some of that oxygen, and the gills in that part of the circuit before it gets to the systemic tissues cannot reoxygenate that blood. And so lower tissue oxygenation is one of the consequences. I should erase that mark. Okay, there we go. So let's look at the circulatory system in a lungfish. So in the lungfish, we basically have the case where these fish have a very expanded air-breathing organ called the lung. And here's the heart down here. And the heart actually has kind of left-right division. And so even though there's just a single chamber, because of various ways that the tissues inside the chamber are oriented, the blood kind of stays on the left or the right side of the heart. And so the left side of the blood goes through the vessels and makes it all the way out to the tissues, and then from the tissues returns to the right side of the heart. And from the right side of the heart, the blood is pumped past the gills and then into the lung, where it's oxygenated, and then returns back to the left side of the heart. So here we have left-right division of the heart in lungfish. Although there's not a left and right heart, the spaces inside of the heart are divided by tissues so that functionally they can separate a left and a right circulatory system. The left circulatory system here is in red, and the left system goes out and into the tissues where the oxygen gets used up and returns to the right side of the heart, which goes to the branchial and to the pulmonary circuit, and then back to the left side of the heart. Okay, so here's an amphibian heart, and I want you to look at this. So here's the amphibian heart. It has a ventricle, and it has a single ventricle, and it has a left atrium over here, and it has a right atrium over here. So this is the right atrium, and the right atrium continues behind this main aorta, or main artery that's leaving the ventricle. And so what is the structural difference about the amphibian heart from an air-breathing fish, the lungfish that we just looked at? And what is the physiological advantage of that? So this is a top hat question. So I want you to look at this picture a little bit and think about it, and then go and answer that question on Top Hat. Okay, so you've answered the question on top hat. If you haven't, pause the video and go do that. So what is the advantage? Of course, the differences are that there are two atria, a left and a right atrium, in the amphibian heart, and the fish heart only has one atrium. However, just like the fish heart, the amphibian heart has a single ventricle, although the ventricle is able to separate oxygenated and deoxygenated blood, and part of it is these folds inside the ventricle that help the blood to flow, so that when it flows out of the right atrium, the blood can be directed to different places than when it flows out of the left atrium. So they have basically two systems in their in the blood that flows out of the heart. They have a pulmonary system where oxygenated blood from the lungs and the skin is directed into the systemic arch. So the systemic arch is this one over here. And then they have deoxygenated blood that's directed to the pulmonary arch, which is over here to the lungs and the skin. So, so they're able to basically, by having these two atria and a ventricle with a number of septations, so this is like a division, including this thing called a spiral fold, they're able to have blood that's flowing to particular places, so that oxygenated blood flows to the muscles and the brain and other tissues, and deoxygenated blood flows to the skin and the lungs for sites of gas exchange. Okay, in reptiles, it starts to get a little more complicated. So this is a non-crocodilian reptile circulatory system. So they have two atria, a left atrium, which is here, and a right atrium, which is here, that structure behind the arteries and the aorta right there. And they have one ventricle, but their ventricle has big muscular folds called a septa. So there's a horizontal septum, which is right here, and there's a vertical septum, which is right there. So these are like parts of the heart muscle that are helping to form chambers, and when the heart is beating, they can make contact with other parts of the heart muscle to basically close off pathways to circulation. And from this, they can have shunts where they can basically, depending on where these septa go, they can bypass where the blood is flowing or which of the pipes the blood is flowing out of. And they can bypass the pulmonary circuit or they can bypass the systemic circuit, depending on what kind of physiological state they're in. And so, for example, turtles have the ability to spend very long times, months, underwater. Of course, turtles breathe air, but they can stay at the bottom of frozen lakes all winter. Turtles can stay in the bottom of oceanic regions for several months without breathing. And so during that time, there's no point for blood to be flowing through the pulmonary circuit, because the pulmonary circuit isn't doing anything since you're not breathing. And so the heart is able to shunt blood just to the systemic circuit. So in a right to left shunt, the deoxygenated blood bypasses the pulmonary circuit and re-enters the systemic circuit. And so that right to left shunt is what happens during breath hold. In a left to right shunt is when pulmonary blood basically goes back into the pulmonary circuit. And so this is what might happen, for example, if there needed to be increased oxygen delivery to the spongy myocardium of the right heart over here. So so this is actually kind of like having a separated left and right system, but the separation has to do with regulation of the shape of the heart muscle through the septa, the horizontal and the vertical septa. And so if we look at what this looks like, for example, in diastole, when the heart is filling, here we have the right atrium is getting filled with blood that's coming in from the main venous system on the right side, and then the left side, blood is getting filled into what the ventricle is divided basically into what is called the cavum arteriosum and the cavum pulmonale. And so the cavum arteriosum, this blood is going to be pumped out during systole through the main systemic arches, the left and the right aortas. So the cavum arteriosum pumps that blood out through the left aorta and the right aorta, and the cavum pulmonale will direct that blood out through the pulmonary arch, for example. And so we can see how the movements of the various septa, for example, aid in the way that the blood is filling and the way that the blood is expelling. So here's the main septa here, the vertical and the horizontal septa that are helping to divide where the blood is flowing out. And so you can imagine it's the septa moved over to here, for example, the cavum arteriosum blood and the cavum pulmonale blood would be shunted through to the systemic arch. In crocodilian reptile circulatory systems, so crocodiles and alligators, it gets even a little more complicated. So here's diastole when the heart is filling, and so here's the left and right ventricle, and the left and the right atrium. And here the heart is more well divided. And so we have a right aorta here and a left aorta there. And there's this spot called the foramen of Panizzi. And this is a connection or an anastomosis between the left and the right aorta. And it allows blood that comes in through the right aorta to go out through the left aorta. There's another anastomosis that happens down here where there's connections between these two blood vessels. And that allows for complicated regulation of where blood is flowing. So for example, if there's a pulmonary to systemic shunt, so over here in late systole with a pulmonary to systemic shunt, what we see is that blood flows out through the both of these aortas, and it goes out through the left and the right aorta, both to the systemic circuit. But if there's in late systole no pulmonary to systemic shunt, then we may see blood flowing out only of the left, only out of the right aorta, and then blood actually flows into the left aorta using this anastomosis down here. And so these are blood vessels, and as we'll see in a little bit, these blood vessels can be regulated by smooth muscle tissue around the blood vessel to open or close the ability for blood to flow through them. And so just like the non-crocodilian reptiles, the shunting aids in selective perfusion of tissues that are in use, so that the heart is not perfusing the entire body of the animal, it's just perfusing the part of the animal that needs the oxygen. And just like the turtles, crocodilian reptiles can also spend a lot of time underwater, in which case there's no sense in circulating blood through the pulmonary circuit. Okay, so bird and mammalian circulatory systems have two complete circuits. They have a systemic circuit and a pulmonary circuit, and they also have a lymphatic system for fluid retention in the circulatory system. So here's kind of a more complicated drawing of how that looks. There's the heart, which is here, and the heart pumps blood out either through the aorta, the systemic circuit, or through the pulmonary artery, through the pulmonary circuit. The blood that gets pumped out of the into the aorta and the systemic circuit goes to the tissues where there are capillary beds. The blood collects in the venous system and is returned to the heart in a deoxygenated state, and then gets pumped out to the pulmonary arteries, goes through the capillaries in the pulmonary, the lung system, collects in the pulmonary veins, and then back, and then that oxygenated blood back in the heart to be pumped out to the systemic circuit again. So here's one way to look at this, where we have the heart and the systemic circuit into the tissues, and then back into the lungs and the pulmonary circuit, and then back to the heart. So that's one way to draw that picture. In terms of the amount of vasculature, the heart itself has about 15% of the blood. The pulmonary circulation circuit has about 12%. The systemic circuit about 16 to 18%. The arterioles and capillaries, the small vessels, have about 5 to 7%, and the veins have about 50%. So most of the fluid in the closed circulatory system is in the venous part of the system, especially in the systemic venous system. And so that's why there's this big balloon, basically. This is where the blood reservoir for the organism is. And of course, the arterial system, this is the blood pressure regulation, as we're going to see. Okay, so here's different types of blood vessels, and just to give you an idea of the differences in the tissues. So here's an arterial vessel over here, and here's the blood. The blood is flowing through this pipe in the middle, and the pipe is lined with an endothelial layer of cells. And in the arterial system, there's an endothelial cell layer. In the venous system over here, there's the same endothelial layer. In the capillary system, there's the same endothelial layer. And then there's also a basement membrane in each case. The capillary is only the endothelium and the basement membrane, so that's all the capillary is, very thin-walled. The venous system has a very small amount of smooth muscle that surrounds the basement membrane. And in the arterial system, there's large elastic components, these internal and external elastic lamina, and then there's a lot of musculature. There's a lot of smooth muscle, and then there's this tunica externa, which is very heavily muscled in the arterial system, but well oxygenated, but not so much in the venous system. So the arteries have a lot of muscle, very thick-walled vessels. The venous system have a little bit of muscle. The capillaries have basically no muscle, although as we're going to see, there sometimes are muscles that are wrapped around the tubes. Draw that a little bit better, like this. They're wrapped around the tubes like that, and then they can, those smooth muscle rings essentially can contract and squeeze down and close off that blood vessel. Okay, so bird and mammal hearts have 4 chambers. They have a left and a right ventricle. They have a left and a right atrium. There's a lot of text here. So there's the left atrium pointing right here. The left ventricle is pointing right here. Here's the right atrium. It's pointing here. And the right ventricle, where's our right ventricle? Well, here's our right ventricle is over here. There's no label for the right ventricle. Anyways, that's the right ventricle here. The muscle tissue is the myocardium, of course. And here are the pipes going into and out of the heart. The pulmonary artery, which is going out of the right ventricle into the pulmonary artery. to the pulmonary system. The left ventricle goes out through the aorta, which is here, the systemic arch. Here's the return to the heart from the lung in the pulmonary veins. And the pulmonary veins go into the left atrium. And here's the systemic veins, which are called the vena cava, or the main vein. And they enter into the right atrium. So there's the superior vena cava, which comes in through the top, and the inferior vena cava, which comes in from the bottom. And they both enter the right atrium here, and they fill the right ventricle. So I want you to draw, and you have to draw in Top Hat. That's what I've learned. Draw the path of blood as a single circuit, like a loop, from the left ventricle back to the left ventricle. So you're going to make an entire circuit of blood, leaves the left ventricle and then returns to the left ventricle. And in your drawing, you're allowed to split the heart into separate pumps. So you can have 2 or 4 or however many separate pumps you need. Include the atria, arteries, and tissues in your drawing. So go to Top Hat and draw that so that you can see basically what this complicated drawing looks like if you draw it in a more simple fashion. And what I mean by a more simple fashion is something, you know, more like what we looked at. Like, I like this one back here, you know, like this is like the kind of simple drawing that I'm talking about. So do that. Okay. So do that and then move on to here, because if you don't do it before you move on, then I'm going to give you the answer right here, which you don't want. You want to actually think it through. Here's a couple of drawings of the bird and mammal heart that show kind of what you might have just drawn. Here's one where the heart is just one pump, and here's one where you've divided the right heart and the left heart. And when you draw the picture like this with the left heart and the right heart, it doesn't look very complicated at all. And so that's part of this lesson, is that there's the right heart that pumps through the pulmonary circuit and the left heart that pumps through the systemic circuit. And so if you can get that, you can understand what's going on. And of course, this is the atrial and the ventricle part of each of the sides of the heart. Okay, moving on. So mammalian cardiac muscle is called a compact cardiac muscle. So there's no sponginess to this muscle. And blood does not get delivered to the heart from, the oxygen doesn't get delivered to the heart muscle from the blood that is flowing through the heart. Instead, there are arteries that are perfusing the heart, and these are called coronary arteries. So here's the coronary artery, and it's flowing over the heart muscle. And here's an actual heart showing the actual coronary arteries. So here are all the blood vessels that are on the outside of the heart muscle. So they're surrounding the heart. And they direct oxygen delivery to the myocardium, or the heart muscle. And of course, that's important. And coronary heart disease, which you've probably heard of, or heart attacks, happen when there's inadequate flow of oxygenated blood to the heart. And so that often happens when, for example, these coronary arteries become clogged. So here is a picture of a coronary artery that is starting to collect cholesterol along the walls, and the cholesterol can form a plaque, and the plaque eventually causes blood vessels to start collecting. And if they collect there in sort of a solid clump, which is called a clot, and blood stops flowing, then of course there's a problem here because there's no oxygen getting to the tissues. And so here over here are some micrographs of what it looks like when there's a artery that doesn't have any blood flowing through it. So this artery just kind of ended, and in a surgery they put a stent in the artery right there. A stent is like a little tube that opens this clot, and now blood flows through again. So if you have a bypass surgery, oftentimes the bypass is bypassing clots that are in these coronary arteries. Okay. Human hearts are aerobically poised. So the human hearts have high ATP demands to sustain the contractile properties of the heart. The adult human heart, so yours and mine, beats about 100,000 times a day and pumps about 2,000 gallons of blood a day. Of course, it's the same blood getting pumped over and over, but 2,000 gallons is a lot, it's a lot of blood. And our hearts utilize aerobic pathways to maximize the ATP yield per oxygen molecule, or this is sometimes called the P to O ratio. And so we use metabolism through oxidative phosphorylation and energy sources that are not particularly oxygen limiting, like the use of fatty acids, glucose, and lactate. And so you're not going to do this part, of course, from my other class. But what about humans that are adapted to high altitudes? Like, for example, the Sherpas that live up in the Himalayas, or the Quechas that live in the Andes. So these are humans that routinely live under low oxygen because they're living at 7,000, 6,000, 5,000, 6,000, 7,000 meters in the very tall mountains. And do you think their heart muscle metabolism would be different? And it turns out that their heart muscles have adapted to lower oxygen. So they have muscles that are in their hearts that are particularly well suited to using carbohydrates. And so they have P to O ratios that are higher when metabolizing glucose than when metabolizing fatty acids. And so they can basically get energy primarily from carbohydrate fuels instead of primarily from fatty acid and lactate fuels like we do. It's less, those fuels are less energy dense, but they're providing ATP under lower oxygen. So when you're up in the Himalayas next and your Sherpa is carrying your pack and is not struggling at all, whereas you're wheezing and you can hardly breathe, and you think that something is wrong with you, there's nothing wrong with you, but the Sherpa physiologically is different than you because their hearts are metabolically poised in a different way than your hearts are. And this is a genetically based change. It's an adaptive change. There's a really nice paper that came out, now 20 years ago or so, that showed that this difference is a genetic difference, that high altitude human populations evolved adaptations in their heart muscles to live under lower oxygen. So no matter how much you train, that's not going to change. Okay, let's talk a little bit about the electrophysiology of cardiac pumping. So as I mentioned very much earlier in this lecture, there's pacemakers that initiate the contraction of muscles by local depolarization of cell membranes. And in the vertebrate heart, it's at a place called the sinoatrial node, or the SA. And so that's right up here. That's in the right atrium. And this is the start of the pacemaker, is that tissue in the sinoatrial node. The sinoatrial node then spreads that electrical connection through non-contractile muscle cells called the bundle of His, and they go down into the ventricles where they cause the ventricles to contract. If we look at the electrocardiogram, this basically shows the depolarization or the voltage of the muscle cells over time. And so what happens first is that the pacemaker starts the atria depolarizing, and so that's this P wave, is when this atrium depolarizes, and it's relatively small because the atria are not that muscly. And then the QRS parts of the ECG, or the electrocardiogram, or sometimes called the EKG, they involve the initiation, the peak, and the hyperpolarization of the ventricular muscles, and then there's the T, which is basically the repolarization of the ventricular muscles. And so this is the signal, and it's basically what we're seeing. First is the atria, and then the ventricles are depolarizing. So here's another look at what's happening. So here's the sinoatrial node, and then here's the atrioventricular node. The atrioventricular bundle is here, and then there's a right and left bundle branch. And so, so these are the non-contractile muscle cells. They're basically just electrically conductive muscle cells that are connected to each other by gap junctions. They don't have actin and myosin, thick and thin filaments. They just are depolarizing and spreading this depolarization. So in these pictures on the bottom, we see the initiation and the spread of the depolarization. So it starts at the SA node, and it spreads throughout the atria. And so up here we see that. We see the sinoatrial node depolarizes, and then the atria depolarize, and then a little bit after that, the atrioventricular node, which is right here, starts its depolarization. And then it takes a little bit of time for that depolarization to spread down through the bundle of His, which is here, and into the Purkinje fibers at the base of the bundles, and then for the ventricular muscle fibers to contract. And then here's milliseconds. So this is half a second from the start to finish. And of course, the heart, at a beat of 120 beats per minute, it's beating twice every second, right? 120 beats per minute is pretty fast. But of course, it can be slower, like 60 would be once a second, and then 180 would be 3 times a second. So you get an idea of what's happening there. This is just a picture to show that what we're measuring is the polarization of the muscle itself. And so when it's in a polarized or depolarized state, we know that's associated with the muscle making its big contraction, as we learned about before. So just again, here's the same picture that we looked at. Here we are in diastole when the heart is filling. Here's where the atria are contracting and blood is being forced into the ventricles. The ventricles then make a contraction. The first thing that happens is that the valves that allow blood to flow out through the main arteries are closed during the isometric ventricular contraction, and that allows pressure to build. And then those valves open and fluid rushes out into the arteries, and then the whole thing starts again. And so we can see this cycle in various places. So here's the ECG. We looked at this before, showing what's happening with the atrial and the ventricular contraction. Here's blood pressure. If we look at blood pressure in the different vessels. So here's blood pressure in the atrium, blood pressure in the ventricles, and blood pressure in the aorta, the main systemic artery. And so during the P wave, we see a little bit of blood pressure build in the atria, and then during the QRS wave, we see a lot of blood pressure build in the ventricle during that process, during that period of ventricular systole or ventricular pumping. And then here's where we see the aortic valve open. So here's the valves down here, the atrioventricular valve, which is closed during ventricular systole, preventing blood from going back into the atrial chambers of the heart. And then here's the aortic valve. And so it's closed for a little while during ventricular systole, and that causes this pressure increase. And then it opens, and the ventricles continue to contract, and there's an even further increase in pressure. And then it closes again as the ventricle stops contracting and the pressure goes back down. And of course, we see this this peak in aortic blood pressure that happens when it closes because there's a little bit of back pressure there. Here's the volume of the ventricle. So the ventricle volume increases when the atria are contracting, forcing blood into the ventricle. It stays constant during the isometric ventricular contraction, which happens here. Then, of course, the volume goes down as it contracts, and then here it fills back up again. And here's the rate of flow. So the rate of flow is 0 out of the ventricle, and then it peaks to about maybe about 500 milliliters per second or half a liter per second, and then it goes back down at the end of the contraction. So that is what happens every time your heart beats. Pretty cool. Here's what that looks like in terms of fluid velocity. So here the colors are the velocity of fluids. And I'm basically showing you the same thing we looked at before. Here's the vena cava, so coming into the right atrium, and we see relatively low fluid movement into the right atrium. And then here's ventricular filling, so now you can still see the vena cava, and there's fluid that flows through the tricuspid valve into the ventricle. And then here's ventricular systole, and we see a huge increase in the velocity of blood that's going out through the aortic valve into the systemic arch. And then in ventricular diastole here, now the ventricle is refilling under relatively low velocity. I think this is just cool. So I'm showing it to you. Okay. Pumps are sometimes defined in something called a work loop. So the work loop is basically the operation of the pump. And so we can think of the pump in terms of the cardiac performance that can be defined on this work loop in two ways. One is the cardiac output. So this is the volume pumped from the ventricle per unit time. And then the other one is the stroke volume, which is the volume pumped from the ventricle per heartbeat. And so this work loop shows us what's happening in the right ventricle, which is down here. This is the work loop for the right ventricle, this kind of darker pink area. And this is the work loop for the left ventricle, this larger pink area that overlaps a little bit with the right ventricle. And when the ventricles are filling, of course, their volume is increasing. So any arrows that are going in this direction is filling, and any arrows going in the direction towards the left is emptying. And this shows us the pressure of the fluid inside the heart. So we see the right ventricle is a relatively low pressure side of the heart. The left ventricle is a higher pressure. It's a left ventricle that is, of course, going out to the systemic arch. And so it has higher pressure. They have relatively the same volume, but the big difference between the two sides of the heart is this pressure difference. And so the left ventricle has the ability to pump blood a lot more strongly and a lot further because of this strong pressure difference. Okay, so when cardiac output has to increase, this happens through two things. One, it happens through changes in the heart rate. So our pulse, for example, goes up when we exercise and we have more cardiac output, but also through changes in stroke volume. Stroke volume, again, is how much blood is pumped for each contraction. So here is a plot of oxygen consumption rate. So metabolic rate is going up to the right. And so we see cardiac output, this red line, goes up during an increase in oxygen consumption. But at some point, heart rate stops going up. An organism reaches its maximum heart rate. Its heart just can't go any faster. But we see stroke volume continues to increase even after heart rate goes up and cardiac output continues to increase. So how is this possible? Actually, I should say cardiac output continues to increase after heart rate plateaus. How is that possible? It's through this increase in stroke volume. And the increase in stroke volume is something called Frank-Starling's law. And it's basically that the myocardial contraction increases with stretch. This is up to a point. But as the filling pressure increases, because the atria are contracting with more force, that stretch of the ventricle causes the ventricle to fill up a little bit more, and it also increases the force of contraction a little bit. And so so this would be like this point right here, would be like that point right there. That's the maximum Frank-Starling stroke volume law that basically says when the heart has more pressure filling it up, it expands more and it contracts more forcefully. You can think of this like a water balloon. If you fill up a water balloon just a little bit, it's kind of flaccid. If you fill it up really a lot, it becomes really hard. And if you open the nozzle to the balloon, the water just squirts out because of the elastic properties of the rubber. Of course, if you fill up the water balloon more than that, it pops, which is bad. And that's really bad for hearts. Hearts don't pop like that, like water balloons do, but just, case in point. Okay, so how are the pacemaker cells controlled, the cells that set the heart rhythm? They have both intrinsic and extrinsic factors that regulate. So intrinsically, they have a spontaneous and rapid depolarization. So everyone's heart's pacemakers are a little bit different, but roughly your spontaneous rate is probably in the 50 to maybe 75 beats per minute, depending on you and your body. And that's just what your pacemaker cells do routinely. But their activity of those pacemaker cells is influenced by neurotransmitters from both the sympathetic and parasympathetic nervous systems. For example, through the parasympathetic nervous systems, the vagus nerves cause bradycardia or slowing down of the pacemakers. So the parasympathetic system releases acetylcholine. Remember this from our neurotransmitter lecture. They release acetylcholine and slow activity of the pacemaker cells by binding to muscarinic receptors that result in increasing membrane potassium conductance, reducing calcium conductance, and that increased potassium conductance keeps the membrane potential near equilibrium for longer and delays depolarization. So, so that slows the heart down. The sympathetic nerves cause tachycardia or faster heart rate, and so they release norepinephrine, which bind to beta-adrenergic receptors on the cell surface that releases adenylate cyclase, increases levels of cyclic AMP, and the cyclic AMP activate cation channels, increasing the rate of depolarization. So, so these pacemaker cells are still the ones that are regulating the frequency of the heartbeat, but they themselves can be regulated by these external neurotransmitters. Okay, so the endocrine system can also do this, and this graph basically shows you some of the mechanisms by which the adrenergic receptors that we see here can activate through G protein coupled adenylate cyclase, which causes cyclic AMP to go up, which activates a protein kinase, which can open calcium channels, which can influence sarcoplasmic reticulum, which can regulate circuit pumps, which can influence the binding of thick and thin filaments. So there's all sorts of ways in which stroke volume is regulated through the hormonal and neurotransmitter mechanisms. In terms of the stretch sensing and the Frank-Starling's law of the heart, we can think of arterial pressure and blood pressure is also being important in regulating the heart. And so this is through baroreceptors. Remember, we talked about the different kinds of mechanoreceptors, and baroreceptors are the ones that sense pressure. So if we start up here and we have an increase in MAP, which is mean arterial pressure right there. The mean arterial pressure can go up, which causes those baroreceptor cells to increase their firing, which causes afferent neurons to send signals to the cardiovascular control system in the medulla of the brain, which causes a decrease in sympathetic output, which causes a decrease in norepinephrine release, which causes the arteriolar smooth muscles, so these are muscles that are surrounding the arteries, to relax, which causes vasodilation, which causes a decrease in peripheral resistance, which causes reduced mean arterial pressure. The less norepinephrine can influence the sinoatrial node, as we just talked about, and can reduce heart rate, which reduces cardiac output. Reduced norepinephrine causes in the ventricular myocardium a decrease in the force of contraction, as we just talked about, through all of these protein kinases, which reduces cardiac output, and all those things reduce mean arterial pressure, and that, of course, causes a negative feedback to the starting condition of increased arterial pressure. The baroreceptors that are the most important in this regulation are in the carotid artery in the neck and in the aorta right at the heart itself. And so blood pressure gets regulated in these manners through baroreceptive responses. Okay, if we look at the entire circulatory system and we look at the velocity of flow and the cross-sectional area, we see they're inversely correlated. So up here is total cross-sectional area of the vasculature. The aorta has very little, arteries have very little, and we see most of the cross-sectional area is in the smallest arteries, the capillaries, and the smallest veins. So the arterioles, the capillaries, and the venules. And then the larger the veins get, the less cross-sectional area. Though notice that this curve has a right shoulder, and that's because the venous system is where most of the blood is stored. And so there's more area in the venous system than there is in the arterial system. If we look at this in terms of blood velocity, what we see is that the velocity is very high in the arterial system, and it drops to almost zero in the capillary system. So at the capillaries, the blood flows very slowly, and that's true in the smallest venous system as well. And then it picks back up in the larger part of the venous system. And so at the capillaries, there's most of our blood, and it's moving very slowly. And this is an important part of the ability for that blood to make exchange with our tissues. And part of the reason for this difference in flow rate has to do with the physics of fluid movement through tubes. So this is just a tube showing that we have aspects of this tube related to the length of the tube, the radius of the tube, the pressure on the inside of the tube, and the pressure at the outside of the tube. And we know that inside tubes, fluids move slowest near the edges of the tube and fastest in the center, and this is because of edge effects or shear effects, which are dependent on viscosity. And how all of these parameters are represented is through something called the Poiseuille equation, which basically says that flow rate is a function of the difference in pressure from the entrance to the exit of the tube times pi to the radius to the fourth power. So this is very important. The tube diameter is really extremely important in setting the flow rate, divided by 8 times the viscosity, this is the viscosity, times the length of the tube. So really it's the radius of the tube that's setting the flow rate, because it's the radius to the fourth power that's dominating this equation. Or tube diameter is really, really important. Okay. So blood pressure, we generally talk about blood pressure in terms of the arterial pressure, and we have the systolic pressure, or the pressure during systole when the heart is contracted, and the diastolic pressure, or the pressure during diastole when the heart is relaxed. And so when you go to the doctor and they measure your blood pressure, it's 2 numbers. the systolic over the diastolic, and that's what that means. The arterial pressure is the same as the heart pressure if they're measured at arteries that are at the same height as the heart. So that's why they take your blood pressure in your arm, in your biceps of your arm, because it's at the same height as your heart, and you do it like sitting up typically, so that they're at the same height. If you were to measure the pressure in a different part of your body, it would be affected by the difference in vertical displacement and the effect of Earth's gravity on the fluids. So that's what's going to be demonstrated. Oh, not yet. In the next slide. So here's a plot of how pressure varies across the vasculature. So here's blood pressure on the Y, and then here's from the heart, basically, is over here, right? And it gets pumped out to the large arteries, the terminal arteries, arterioles, capillaries, venules, and veins. And in the beginning, we really can see that signature of systolic and diastolic pressure, the difference in systole to diastole in every heartbeat. That's what these zigzags are here. And we basically have blood that's moving in pulsatile waves. So we have a wave of blood that goes forward, goes back, goes forward, goes back, goes forward, goes back, as the heart is contracting and relaxing. But eventually that smooths out so that by the time our blood is flowing through the venous system, it's a low pressure, just continuous flow. Of course, you've seen this if you've ever watched like a horror movie and someone has an artery cut in the horror movie and you see blood squirting out, right? So that's what's happening if there's a cut in the circulatory system up here. But probably all of us have experienced a cut in our bodies at one time or another where the blood just kind of flows out. And we're dealing with probably a cut in the capillary system here. Hopefully you haven't cut a venule or a vein, because you could lose a lot of blood if you did that. But probably just cut like your finger or something like that, and the blood just kind of flowed out gently. That's because it was here in your body where there's low pressure and laminar flow of the blood. Okay, so getting back to the effect of gravity on blood pressure. So now we've taken this fluid column and we've turned it up, and so now we have, in addition to the length and the radius and the difference in pressure from one side of the tube to the other, we also have the effect of gravity. So we have the pressure, a difference in height, and then the pressure down below is equal to that pressure plus the density plus the effect of gravity plus the height. And so the heart, if it's going to pump blood up, has to pump against those additional forces. And for most humans, the maximum vertical pumping distance, based on the size of the heart to where there would be zero pressure left, is about 1.5 meters. So that is to say, if you raised your hand over where your heart was, and your hand was more than about 1.5 meters above your heart, it would be very difficult for your heart to pump any blood into your hand, and your hand would basically become deoxygenated. Most of us don't have arms that are that long, but for very, very tall people, I guess this could be a problem, that if they raise their hands above their head for a very long time, they could lose blood flow in their hands. Similarly, the pressure in your feet, which is quite a bit below the heart, can be very much higher than the blood pressure at the heart in the arterial system, because of the effect of gravity on this vertical column of fluid through the systemic arteries that are going down into the feet. That's probably why we like to put our feet up when we're sitting. And this brings into question long-necked mammals. So here's a giraffe. Of course, a giraffe has a very long neck, right? The giraffe's head is maybe 5 meters tall, and the giraffe's heart is maybe about 2 meters tall. So the giraffe's head may be 3 meters above its heart. And so this poses a big circulatory challenge for giraffes. And their adaptations are that, first of all, they have a very large heart. So giraffe hearts weigh about 24 pounds, and they can pump about 16 gallons of blood per minute. Giraffes also have polycythemia, so they have about twice the number of red blood cells per volume as we do, and they have the highest recorded systolic pressure of any animal at about 220 millimeters of mercury. That's about twice what the healthy human systolic pressure is. They also have rete mirabilia and blood vessels at the base of the brain. So this allows blood storage near the brain and prevents blood for basically flooding the brain if the giraffe happens to bend its head down to drink. And the giraffe also has a lot of one-way valves in its neck to prevent the blood from flowing back to the brain on the way to the heart when the giraffe has its head in the down position. And so again, the giraffe has to pump the blood from its heart all the way to the top of its head, which, maybe several meters. And then sometimes the giraffe's head is below its heart when it's drinking, for example, because water doesn't come 6 meters high off the ground. Water comes at the ground. And so the giraffe basically has issues when it does this. When the giraffe is standing up, there's vasoconstriction of vessels in the lower body to prevent blood from pooling up in its legs. And when the giraffe lowers its head to drink, it has big differences in the aortic pressure, and the one-way valves and rete mirabilia now prevent blood from flowing down into the head and basically causing, massive swelling and blood vessel ruptures and stuff like that in the giraffe's head. So that brought me to the logical continuation of what circulatory system adaptations might sauropod dinosaurs have had. So here's us, right, Homo sapiens. Here's a giraffe. That's a big, tall giraffe. And then here's some dinosaurs, sauropod dinosaurs. And this includes Brachiosaurus and Saurosospedian, very, very large dinosaurs. And it's thought that most of these very large dinosaurs had their bodies in a horizontal position like this, so that their heads were about the same height as their hearts, like Apatosaurus, for example, did this. And so this does not present any kind of a blood pressure challenge because there's no effect of gravity on this blood. It's vertically at the same spot as the heart. But based on the structures of bones, it's thought that Brachiosaurus had elevated heads, that this dinosaur actually had a head that went way up like this. It could have been as much as 8 meters above the heart. And so if you just take the giraffe and extrapolate to what these dinosaurs might have had, they could have had a heart that weighed 400 kilograms. That's a big heart. They could have had huge blood pressure, like 600 millimeters of mercury. So like 3 times what the giraffe has. And very, very huge muscular blood vessels, like really wide arteries, really strong arteries, and probably a lot of one-way valves in the arteries and veins to prevent this dinosaur from having like a blood embolism in its brain when it bent its head down to take a drink of water. By the way, these dinosaurs are about 30 meters long, so about almost 100 feet long, and lived about 100 million years ago. So there you go, paleophysiology. Okay, so the capillaries are where the blood and tissue exchange occurs. And so capillaries are very small. And actually, here's a picture of a capillary, and each one of these little dark things is an individual red blood cell. And so we can see the red blood cells are basically just about the same size of the capillaries, and they're squeezing through kind of like in single file. And so there's a lot of potential for diffusion between the capillary and the surrounding tissue because these distances are very small. diffusion flow between the capillary and the tissues is aided by blood pressure as well as osmotic pressure. So at the arterial end of the capillary, there's elevated blood pressure, right? Because the flow is going this way through the capillary, and so the pressure is higher on the arterial end. And so that pressure causes filtration out of the capillary. And so what we're looking at down here in this graph is pressure versus capillary length from the arterial to the venous end. And so we see that the pressure of the blood causing filtration to happen. And then at some point, the macromolecules that don't flow out of the blood become more concentrated in the blood than the tissue. And then there's an osmotic pressure that happens where the arterial pressure is actually lower than the osmotic pressure, and that causes uptake of materials at the venous end of the capillary into the blood vessel. And so as stuff filters out of the blood vessel, the stuff that remains becomes more concentrated, causing an osmotic challenge that results in uptake of stuff into the capillary in this area too. So there's an osmotic pressure gradient that exists at the venous end, which is how the capillary fills with fluids. Pretty cool. Capillaries are covered with contractile cells called pericytes that help to direct flow. And so I mentioned that there's smooth muscle that surrounds the pipes in the circulatory system. Remember, smooth muscle are muscle cells that wrap around tubes and regulate the tubes. And so we see here at the arterial side of a capillary bed, there's all sorts of smooth muscle cells that wrap around. These are post-capillary sphincters. There's all sorts of nerves that innervate these cells. Pericytes are individual smooth muscle cells that are wrapping around capillaries, and the nerves are basically regulating the contraction of these cells, which regulate which of these pipes is open and available for blood to flow through. And there's smooth muscle cells wrapped around the arterial as well as the venous side of the capillary bed. And so this is important for regulating the flow of blood through the capillary beds. It's also important for erectile tissues, which we might get to in a sec. Here's a whole bunch of different factors that are influencing vasoconstriction and vasodilation. A number of factors that cause vasoconstriction, and a number of factors that cause vasodilation. And we're going to talk about this one called nitric oxide. So this table is here for your reference. So nitric oxide is a really important molecule. It causes vasodilation or increased blood flow into capillary beds. It's produced as a function of arginine degradation. So here's the amino acid arginine, and nitric oxide synthase degrades arginine and makes something called citrulline as well as nitric oxide. And nitric oxide, this is produced in the arterial cells on the inner surface of the aorta in those endothelial cells. And the nitric oxide activates guanylate cyclase, and guanylate cyclase makes cyclic GMP from GTP, and the cyclic GMP can regulate the processes involved in contraction and relaxation. So it can regulate the protein kinase that regulates the phosphorylation of myosin to myosin phosphate in smooth muscle, which causes an increase in relaxation. And it also can inhibit the phospholipase C enzyme that causes an increase in calcium. And so this causes a decrease in contraction. So it regulates, positively regulates relaxation. it negatively regulates contraction, and it also enhances potassium flux through the membrane by regulating a potassium channel, which also causes the smooth muscle cell to relax or to have a less likely contraction. So you might have heard of Viagra, for example. It increases nitric oxide's effect by blocking the enzyme that degrades cyclic GMP. So Viagra basically helps this cyclic GMP to stay around longer. That enzyme is called cyclic GMP phosphodiesterase, and it's cyclic GMP enzyme number 5 that is only found, well, not only, but is primarily found in the penis smooth muscle. So if cyclic GMP hangs around for longer in this penis smooth muscle, that means that there is more blood flow into the penis, which means that the capillaries are more full with blood, which means the erection happens and lasts longer. So Viagra is not totally specific, however, and has effects in other parts of the body, including kidney cells, and may actually affect kidney function. But in case you were wondering how Viagra works, there you go. It's a part of the muscle, the regulation of smooth muscle in capillary beds. There are different types of capillaries. Some capillaries are continuous. These capillaries are the least permeable. They have some very small clefts. So here's the capillary, right? Here's the endothelial tissue. There can be a cleft between the endothelial cells, and the basement membranes are entirely intact. So diffusion is very slow out of these continuous capillaries. And these are the types of capillaries that we see in tissues like muscle, neurons, lungs, connective tissue, exocrine glands, basically where the blood has to stay in the capillary. Fenestrated capillaries have a little more permeability. There's more space in between the endothelial cells in fenestrated capillaries. In these pores, the basement membrane is intact, however, and these are capillaries that have an intermediate level of permeability. For example, the renal system, which we'll learn about next in 2 weeks, the intestinal system, and the endocrine glands. And then the most permeable type of capillary is called a sinusoidal capillary. And here there's basically a gap between the two endothelial cells, as well as an opening in the basement membrane. And the blood can come out of the capillary. Maybe not the red blood cells, but fluids can flow right out of the capillary here. And this is what happens in some of the tissues where there's the most exchange between the capillary and the tissue, like in the liver, the bone marrow, spleen, lymph nodes, and the adrenal cortex of the kidney, and so, or above the kidney. So, so these sinusoidal capillaries are the most permeable. Okay. To finish up, the pulmonary system, it's a very low pressure system, as we saw in that work loop. And the key feature of the pulmonary system is that there's no fluid exchange in the lungs. So if fluids start to accumulate in the lungs because fluids are coming out of the pulmonary capillary beds, this is something called a pulmonary edema, or fluid in the lungs. It's a very serious medical condition, and it's caused by dysregulation of the cardiovascular system, often caused by clotting. And this is a very serious medical condition that you do not want to have. And then a little bit about how muscular contraction is involved in moving blood back to the heart from the venous system. Of course, the blood in the veins is under low pressure, and there are valves in the venous system, one-way valves, that are basically flaps of muscle inside the veins that allow fluid to move in one direction but not the other direction. So you can make a one-way valve with your hands, right, where they can't move in one direction but they can move in another direction. And that's what's happening inside the blood vessels. And as skeletal muscle goes through cycles of contraction and relaxation, they also squeeze on the blood vessels, and they help to pressurize the fluid. And of course, the fluid only moves in the direction that is available based on the one-way valves. So skeletal muscle is important. The movement of skeletal muscle during breathing or the respiratory pumps is also important because this helps to fill the vena cava and the other aspects of moving blood from the thoracic cavity back into the heart. So if you're on a long airplane flight, for example, it's really important to move around, to get up and move around, because if you don't move your skeletal muscle enough, you don't help your body to get the blood back to the heart, and you can start to have a lot of swelling in your extremities, which is also called an edema. It's not as dangerous to have really swollen feet as it is to have fluid in your lungs, but it still can be unpleasant. So if you've ever gotten off an airplane flight and your feet feel swollen, it's because you didn't have enough movement of your skeletal muscle in your legs and your feet to help pump that blood back to your heart. Okay, so I'm going to stop this first part of the lecture there, and I hope you enjoyed it.